Adventures in the Kingdom of Cummings. What a fantastic beginning to my final year as a teacher in the public school system of Georgia. You North Hall young'uns make me proud to be here. Thanks for giggling when I was silly and for showing awe when I was awesome. This year is going to be magical. Let's do it all. Welcome to the Kingdom of Cummings. Day 179. Colorful butterflies were created in the kingdom today, and the Hocus Pocus slab was delivered. Except for messing up the lockers, it was a very decent day for me. What was your high, low, and funny for the day? Day 178 was a fun day. We played with magic sand and learned about the Tree of Life, Miss C style. I loved the video of black-footed ferrets in the kingdom. ASAP Science explains why the egg came first. Yep, it was a good day. Day 177. The Wump World and the Dig, my favorite childhood story and my favorite classroom project. Thanks for drawing wumps for the first time. They were intriguing. Lots of origami frogs too. So what did you learn about communication today? Day 176. Today's Trojan support time was an adjustment. I think once we get into the swing of things, it will be useful and fun. You guys have any good reading ideas? I have plenty of science tricks up my sleeve and stories to tell. We had winners in the Kingdom of Cummings today. Rosa in first period, Jaron in second, Ruby in fifth, and Darren in sixth period. Their estimate of the length of the sidewalk in meters was most accurate. Kudos. Journals today too. I would like to keep them for you so they won't get lost. Day 175. Using the microscope is so much fun to me. Seeing all those little protozoans, algae, and tiny animals make me realize how diverse life on Earth really is. I will put together a small video clip of the organisms we saw today. I'll show it on Tuesday. Ms. Williams will be my sub on Monday. I have to go to the doctor for a routine checkup. Nothing of concern, just the dermatologist. Day 174. Today we analyzed a dichotomous key. It classified some of my hyperglycopods. We were able to key out Zorgeister and your Stugus. We also identified some of the protists we saw using a key. Day 173. I love reading student journals. So today, we worked on making our journals easy to identify and a little more personal. We practiced making proper journal entries with a title, the date, and data that is easy to interpret. First and second answered conclusion questions regarding the lab, measuring with estimates. Ms. Cathcart talked to the girls about discipline, then the boys. Fifth period was very thoughtful and devised a way to solve the problem of time management. Sixth period had extra time to observe protists using the microscope. Great stuff today. Day 172. I enjoyed the lesson today. Observation, inference, and prediction using the You'll Never Guess canisters, Skittles, and the PowerPoint with BC comic strips. They were very effective. What did you think was the strangest thing in a canister? We also got some tadpoles today. And the thunder fifth period was intense. Day 171. Fun day today. TST was spent reading, making flying dragons, looking at pond water, finishing homework, and all sorts of stuff. I did compliance. We reviewed the 10 science process skills today. The lab today was successful in 2 out of 4 classes. First period could not manage the lab appropriately, so the bouncy balls were taken up. Second did a much better job of collecting data and recording it in their journals. Nice. Fifth period had a couple of students who went to timeout for not following directions. And sixth period did such a fine job that each student was awarded a bouncy ball to take home. Yay, class! So what variables did we hold constant in the lab today? Day 170. Today we got to know each other a little better by choosing a favorite personality picture. Several students wanted to read the description that accompanied the personality test. And alas, today was the last day to use my daddy's lead casing at school. 8 pounds, dense, 11.3 grams per milliliter to be precise. Winners in the kingdom were Shay, Emily, Maggie, and Darren. Way to go, young people. At the end of the day, I bequeathed a piece of lead to Ms. Ellis and took the rest home. I love labs where precision counts. And those students who commented on my blog got a response today. I like this blog thing. Day 169. Got my toes in the sand. There's a song there. The beach. Sand. I love the stuff. I've been collecting it for years. Today we observed sand and tried to determine the beach or desert from which it came. Indiana Cummings lost a research assistant on Tortuguero Beach and wants to know who done it. Mysterious sand was discovered at the scene of the crime, and forensic scientists came in to analyze sand from each of the suspects. Whose story will be most convincing and fun to read? Day 168. Wow, a writing day. We took sand analysis and turned it into murder mysteries. Included Cumberland beach pictures and video clips. We had a short show and tell. We learned what fulgurites are and what saddleback caterpillars can do. Day 167. How about those butterflies in the kingdom today? That was totally fun. And we got some exercise. Each class worked together to ensure good video and photography, that all the butterflies' needs were met, and 2 winners emerged by the end of the period. What was your favorite task? and who won each period? Ooh, lots of comments on day 167. Day 166 was picture day. We were in the computer lab doing research. We researched plankton ideas for the lab tomorrow called Sinking Slowly. Yes, SpongeBob's plankton is okay, but the winner will be the model that takes the longest time to sink through the water in an aquarium. 1 minute and 26 seconds is the time to beat. Day 165. Plankton models. Yay! About 50% of first and second came prepared today, and all but 2 in 5th and 6th. Good job, afternoon classes. Kristen and Michaela won first. Elena and Noah won second. Kyler's sank the slowest in 5th. And Aurora and Levi's won 6th. What makes a good plankton model? Day 164. Water spheres, water molecules, were awarded to the first person in each class who knew what the next unit will be. They remembered the syllabus. Life on Earth, David Attenborough, Changes Through Time. I created this unit 29 years ago based on the award-winning book and video called Life on Earth. It chronicles increasing complexity, classification, and the characteristics of life over a 6-week span of time. My goal is to obliterate ignorance and enlighten the eager young minds in my classroom. So, where did you come from? Day 163. Amino acids, then proteins, then DNA, and a cell membrane undergo fission, and you get the first cells, prokaryotes. And through endosymbiosis, eukaryotes. Phew! We learned some life on Earth today. Doesn't it make sense that the building blocks had to be assembled before the house or the cell could be built? Tomorrow, David Attenborough. Day 162. David Attenborough, my favorite natural history teacher, published Life on Earth in 1979, the year I graduated from high school. In Chapter 1, he describes Darwin's research in South America and on the Galapagos Islands. He explains how natural selection works through variations within a species over time. Simply eloquent. Then he describes the increasing complexity of life leading to the first animals on Earth. What element is fundamental to the building blocks of life? Day 161. First Life by David Attenborough was aired in 2010. In this series, Sir David travels 40,000 miles to show us evidence of snowball Earth, fossils of Charnia, Fractophusis, Dickinsonia, Kimberellia, Sprigina, and Funisia. He talks with expert paleontologists. The series also uses cutting-edge CGI to recreate these early animals. How did each of these fossil animals provide evidence of increasing complexity? I included several links to the series and NPR. Day 160. The Cambrian Explosion was an event in life history that began 542 million years ago. During that period, all modern phyla of animals appear in the fossil record. The most notable fossil quarry is in Canada and consists of Burgess Shale. It is a popular site for evotourism. Your creature creations were adorable and better than any for sale on the internet. Thanks for the photo ops. I created a new slideshow for you. Several comments. Day 159. Life on Earth. Arthropods. Incredibly successful. They have segmented bodies, an exoskeleton, and jointed legs. Critter Hide and Seek is going to be on Friday. My expectations are high. We also studied the increasing complexity of plants, too. Day 158. We got so much done today. We reviewed old blog posts, watched a PowerPoint about natural selection, played a peppered moth simulation game. We watched an HHMI video clip about pocket mice and natural selection. We did Life on Earth Chapter 5 about fish. And finally, we drew carboniferous trees in our journals. So, why are the Cambrian Explosion and the Carboniferous period significant in the history of life on Earth? Day 157. Trilobites. Fossil arthropods that were abundant and diverse during the Cambrian period. Why did they evolve such a wide variety of eyes? Hostways. Beautiful picture of a trilobite from 2013. Day 156 was also a day that Dr. Rayburn did an observation. Day 156. Critter Hide and Seek is a wonderful activity about arthropods, animals with jointed legs, an exoskeleton, and segmented bodies. Critter qualifications included using 5 man-made materials to build your models, using characteristics of one of the 4 classes of arthropods: Myriapoda, Arachnida, Insecta, or Crustacea. The winners are pictured in the September slideshow. Some comments. Day 155. Today, we watched our Critter Hide and Seek photo story with pictures dating all the way back to 1994. We took notes on the PowerPoint presentation called Changes Through Time all the way to slide 68, the one about Pacaya, which is the ancestor to the vertebrates. Many of you are very good note takers, which is a great skill to have. Now, what were some of the major events in the history of life on Earth? Several comments. 16, wow. Day 154. Flashy Fish was so much fun, it had me doing cartwheels in the hallway, in my flashy oriental jacket to boot. Did I point my toes? That was from my old ballet days. So, what pushes towards flashiness and drabness? How did this activity demonstrate natural selection? Day 153. From Pikaia to fish, amphibians, and reptiles, life on Earth throughout the Paleozoic and Mesozoic eras evolved. Noteworthy fossils include Tiktaalik, Archaeopteryx, and Thrinaxodon. Magnolias are a plant family representing transition between cone plants and flowering plants. We played the game in the kingdom today. We were salmon this time. I think getting up the waterfall was the most difficult part of the simulation. That was when you had to hang for 5 seconds. Not sure everybody tried. The only for sure winner today was Raoul. Day 152. What a nice day today. We learned about dinosaurs and how frogs take care of their eggs. That was Life on Earth, chapter number 6. We began on our dino data charts and will finish tomorrow. At the end of the day, Chris delivered Coach Hicks's dino data from 1997. The oldest dino data charts date back to 1994. I have a whole book. Day 151. We watched Life on Earth, Chapter 7, called Victors of Dry Land, and completed our dino data charts. Since lots were turned in without a name on them, which was partly my fault, it was suggested that this be a bonus grade. The 100s go to Jenna Brock, Sinessa Burns, Riley Freeman, Hope Miller, Ruby Espinoza, Darren Devane, and William Townsend. Great creations. Who remembers some of the cool adaptations of the class of chordates called reptiles? And who can tell me what hydrocaudimaedon means? Or Lophodidontoxenognathus. Those are some cool names. Day 150. What a day was today. First, Ms. Ellis was out, and she's never out. So we helped the sub get ready during homeroom. We went through 80 slides and took notes to the best of our ability. We had to evacuate during 5th period. Teachers sat with their 5th period classes at the high school stadium, allowing students to use the restroom and get water. Some students who were heat intolerant went into the high school gym. One student collapsed due to a pre-existing condition. Miss Jennifer flew to her aid and took care of her. Unbeknownst at the time, Darren Brown had offered to bring me a cup of water, which he did. It arrived at the same time that the faint student was stretched out, so that water got redirected to her. Thank you, Darren, for that random act of kindness. I missed my 6th period today. See you tomorrow. Day 149. 1st, 2nd, and 5th worked on slotted notes, while 6th watched the PowerPoint slides that were missed yesterday. Boy, you guys ask some smart questions. I added several links to tomorrow and Thursday's calendar for further research. This gives you proof, verifiable evidence. Good science stuff. Day 148. Fashion a fish or make a mammal. Y'all made some really cute ones that I'll be able to use as bookmark art. Ah, yes. I will be selling color-changing bookmarks and memo pads for $1 with artwork on them. They are cool. So what are the major characteristics of fish, amphibians, reptiles, birds, and mammals? Day 147. We busted some brain today. How to decipher answers from an outline and choose the one best answer for the review handout. Tough stuff, kiddos. Some of you are already good at that. Tomorrow is National Black-Footed Ferret Day. I'm so excited. They're my favorite animal. And we won the pep rally. Yay! Day 146. September the 26th. National Black-Footed Ferret Day. My favorite mammal, also an endangered species, a predator of prairie dogs, and a muse for artwork. I could go on and on. Check out the links on Friday's calendar to learn more about these fascinating creatures. 5th and 6th periods got to be black-footed ferrets in the kingdom. See pictures in October's slideshow. September's slideshow reached the picture limit. Day 145. Students from the University of Georgia came to visit first period, and they brought examples of a couple of classes of arthropods, both arachnids and insects. They have an exoskeleton, jointed legs, and segmented bodies. Literally, jointed foot. Can you remember the characteristics of these two classes of arthropods? What makes them different? And today was the first time I ever let a tarantula crawl on me. Video evidence. Thank you. Day 144. Food, the ultimate motivator. 95% of you finished your review handout and earned a stamp and a smackerel. Quote, a taste or very small amount, end quote, from Winnie the Pooh. Nice work. Everyone should now have a syllabus. Tree of Life and Chordate handout, Life on Earth slotted notes, PowerPoint notes, and the review for this unit in your notebook. All these will help you prepare for the unit test next week. Several comments. Day 143. Yes, the Media Center, a place to kick back and enjoy technology. Some of you used your tablets, while others used cell phones or the Media Center computers to access USA Test Prep and my webpage to practice answering test questions. Ms. Mabry and Ms. Sapp were spectacular hosts, accommodating our every need. Several chose to work with an old hard copy of the test, utilizing the comfy couches and clipboards. Caleb Chapman brought in an inquisitive stick bug, of which we made a short video documentary. Fun day today. Day 142. Today, we wove a food web of the North Hall Kingdom. Many of you were producers. Several were primary consumers or herbivores. Even fewer were secondary consumers or carnivores. And only one of you was a tertiary consumer. And you always died for the fungus to decompose. What a great opportunity to teach energy flow through the environment. What part did you play? Day 141. What a great day for review. 2nd, 5th, and 6th, you guys outdid yourselves, answering questions, asking questions, and taking notes on the PowerPoint. Bien pour vous. First period had some difficulty getting along with each other. I have many strategies for dealing with this. Please, treat each other nicely so I won't have to utilize them. Big class, lots of personalities, This is an excellent reason to learn proper social skills. Remember, guidance on Monday, test on Tuesday. Plan and prepare. Day 140. Georgia College 411 in the 6th grade computer lab. I learned about career opportunities I'd never heard of, such as dramaturge. Mrs. Hales and Mrs. Henderson were very effective teachers, and everyone saved to their portfolio. What a nice lesson. Tomorrow, test over life on Earth. Study tonight. Day 139. A 50 multiple choice question test over life on Earth. The 3 top scores were achieved by Sanessa, she made a 98, Emily, who made a 98, and Autumn, who made a 96. And I am also very proud of Addie, Georgia, John, Kent, Noah, Darren, Trevor, and David. You all exhibit great skills in observation and an exemplary work ethic. Tomorrow, popcorn and a movie. Day 138. Some folks tied up loose ends by finishing their tests, while others enjoyed Cummings videos with popcorn in their tummies. A few folks were able to decipher their names using DNA codes. The end of the 9 weeks is near. Fall in the kingdom of Cummings continues. We also got an email about being a little noisy that day. Day 137. Journal topic: Increasing complexity of life on Earth. We watched several videos from notable scientists, including Carl Sagan, David Attenborough, and Neil deGrasse Tyson. I shared current averages with you individually, and we watched some funny clips like Evolution on Speed and Homer Simpson's Evolution. Tomorrow, Ms. Goldman will share some Planet Earth clips in the morning. They are in high definition. And I hope y'all enjoy 7th grade reward day in the afternoon. Day 136. I missed you guys on Friday. I hope all went well. At my date with the dermatologist, Davina dug deeper to determine the dimensions of a small basal cell carcinoma on my dorsal side. Ha, good use of alliteration, huh? The margins are clear. Good reason to wear sunscreen throughout your life, so you won't have so many dates with the skin doctor. The Kaolin Festival in Sandersville was fun, and this is a picture of my first skin spot that was frozen with liquid nitrogen going through the healing process. Day 135. Stormy weather prompted me to share this video as a class opener. It was one that I uploaded to YouTube about a lightning storm, 2009. Captured video frames or snapshots are really cool. We went over the characteristics of life PowerPoint and took notes over the building blocks. And the 3D cell model guidelines were handed out and discussed. Cell models are going to be due on Halloween, Friday, October 31st. We will plan for a grand celebration. Day 134. Celebration videos with music to kick off the day. We took notes on PowerPoint slides through 50. I uploaded 2 PowerPoints to Labs and Reviews for you: the characteristics of life and the chemistry of life. Both are down under related files. Sorry I missed 5th and 6th, but vertical planning means I got to share with 6th and 8th grade science teachers. I also uploaded tomorrow's lesson to YouTube. It's called Just Passing By, about a water molecule in the kingdom of Cummings. Day 133. We had a rough morning. T-squared is not a good time to build cell models. They will need to be made at home. The due date is October the 31st. Just Passing By and A Water Molecule in the Kingdom were presented today. They chronicled students writing about the movement of a water molecule in the kingdom. The original activity came from Project WET, and many adaptations have been published since then, which was in the 1980s. I included science and social studies standards as well. So, who can tell me where the Mekong River is, and what river is dammed by the Three Gorges Dam? Where can thermophilic bacteria be found? How about colorful diatoms? Day 132, a festive day at North Hall. The homecoming parade was inspirational, and the lunch that I had with the 7th grade teachers gave me a sense of belonging. Ms. Neville drove the science teachers in her Highlander. It was a nice ride. Mr. Party was yum. Dr. Fish was punny. And in class, we made DNA bracelets. 11 comments today. Day 131. I enjoyed using the new projector with all the colors and sizes for the pens, unlike last Thursday during T-squared. Thanks for all the kind remarks and love wishes. In the computer lab, morning classes did an exceptional job filling out the cell chart and labeling the plant cell. Only one student didn't quite get done out of 58. Some of 5th period decided to be silly, and 6th period did a bang-up job as usual. What are you planning on using for your cell models? Mitochondria. A few comments. Day 130, October the 21st, was a day I had to go visit the principal's office. Day 130. The focus was on DNA. We watched the DNA rap. We sang the DNA song, took some PowerPoint notes on DNA, and watched a visualization of DNA, replication, transcription, and translation. That was cool. The Inner Life of a Cell, put to Led Zeppelin's Stairway to Heaven, had over 360 views on YouTube. You know, it's a flagship rock ballad, Stairway to Heaven. The best notes of the day were Chad's. Nice job. Couple of comments. Day 129. We built models of DNA. Some students went beyond the standard directions and built DNA based on their name sequence from the DNA alias activity. Kudos to Isaac, Sinessa, Addie, David, and Maddie. Reading and following directions enables you all to create your own personal DNA sequence. 6th period was able to put everyone's DNA together to create one big happy family. We made a video. Cool. Several comments. Day 128. We completed the DNA lab today by drawing our name in DNA code. The DNA notes from the audio portion of yesterday's lesson were helpful in answering the conclusion questions. Maddie Harrison's unzipped DNA picture is very impressive. I can't wait to take it in Photoshop. Day 127. I hope all went well on Friday. The crossword was a good way to review. Your DNA pictures looked nice. Some were dead-on accurate. Addie, Sinessa, and Jenna's were my favorite first period. Yare, Elena, and Chase's were my favorites in second. Darren, Trevor, and Chastity's were the best ones in fifth. And in sixth, I liked Maddie's, Saul's, Autumn's, and Maddie's DNA. Very nice work, you guys. Day 126. Cell parts and their function was the focus. The computer lab was fun. I heard lots of sound and music. I also saw lots of research and curiosity about cell models. Party planning has begun. Please continue to work on your models for Friday. Day 125. Cell processes and demonstrations were conducted today. We reviewed the Chemistry of Life PowerPoint and added a few new slides. We looked at Ms. C Does Magic on YouTube, and Ms. C does an egg experiment in the classroom. Several of you experimented with the dialysis tubing containing turmeric dissolved in alcohol using the ammonia and citric acid solutions. It was a wow kind of day. Day 124. Investigating cells, the lab. Second and sixth did a phenomenal job. We got some great pictures. I'm inquiring about the dark spots on the Eladia. We'll see. Day 123. Music is relaxing to me. You all seem to enjoy the PowerPoint with the 4 varied songs too. Focus today, mitosis, is the process of asexual cell division of a nucleus. You created flipbooks today. Addie, Kyler, and Sam struck me as special keepers. Yay! Happy birthday, Autumn. I love my birthday song to kids. Tomorrow, our celebration. Come prepared. Day 122. Our celebration was a blast. Only a few students had an alternative assignment. Many of you were able to tell me all about the cell parts and their function. Bravo! I thought the tastiest cake was Faith Chandler's, and the prettiest one was Daniela's. It was a double-decker cake, chocolate and vanilla layers. You can download any pictures you like from my slideshows by clicking on the thumbnail, making it larger, then right-click to save as image. I like being able to share. Day 121. All about cells. Slideshow about our Celebration Friday. A taped lesson. Endosymbiosis. Mitosis. And a celebration video. All good for review. Here's a YouTube link to the final video, which includes the best of the best. It's on my YouTube channel. And it's good. Day 120. I thought today was very productive. Most of you now have proof, verifiable data regarding cells. Keep up with your charts and reviews. Tomorrow, more review. Test Friday. I so enjoyed the eager young minds who work like Trojans to accomplish the task at hand. water spheres for rewards. They're hydrophilic, like the phosphate heads of the cell membrane. Science rocks. Day 119. T-squared time test review was very successful, as evidenced by Cade and Lydia. They knew all the answers during class, smarty pants. The cells presentation was highly motivating and visually pleasing. I'm glad you liked it. Maybe we could add some of the test questions to make it even better. Test tomorrow. If this is a plant cell, can you name all the organelles shown? Several comments for answers. Day 118. Our Veterans Day assembly was the focus of my day. The band and chorus were uplifting and made me proud to be a Trojan. I managed to record the band, but when the chorus sang, my battery became exhausted. Jay recorded it for Miss Thayer, thankfully. My heart beamed for all of you. Day 117. New unit, heredity. I introduced a PowerPoint. Lots of discussion regarding Nick Vujicic. He was a guest speaker at North Hall Middle School on 9/26/2008. He's married now and has a son, Kiyoshi. What a man. Day 116. Watch genetics videos about disorders and medicine and technology. What choices do you have about your genetic future? Day 115. Investigating inherited traits, also known as the Making Babies activity. The commentary during this activity was quite interesting in regards to diversity and freedom of choice. I loved the names of some of your offspring: Sharkeisha, Larissa, Annabelle, Carissa, and George. Where did your traits come from? Day 114. Investigating inherited traits. We did the vocabulary, and we did an activity that was an inventory of my traits. I edited some wrestling videos and showed some interesting traits from former NHMS students. So, what's a hitchhiker's thumb? A widow's peak? A cleft chin? Which way does your hair whirl? And can you roll your tongue? Day 113. Spongebob Genetics. Only a small percentage of you figured out the genetics of Bikini Bottom. Solving Punnett squares requires a little reading, like word problems in math. Look at this diagram. Whose alleles are represented here? Day 112. A rainy Monday. Good day to finish worksheets and tie up loose ends. My webpage pulls it all together and allows for organization and reflection. What are alleles? Why must 2 coins be tossed for each trait? I got some comments with some answers. Day 111. We reviewed the Investigating Inherited Traits handout and practiced some more Punnett squares. Some classes got to see some old gene role play skits from former students. We watched the genetics rap and a couple of clips about tissue engineering. Day 110. The Human Genome video clips. A1 Milano, telomerase, P53, free radicals, and mitochondrial DNA were all discussed. Why do mitochondria have their own DNA? We watched some more gene role plays too. That day, Ms. Benton came in and did an observation. Day 109. So, could the dog be called Spot? Why? Give an example of its genotype and phenotype. We decorated the new board well. Day 108. Research. Lots of choices regarding genetics. Some choices were very close to home. Some of you shared your documents and PowerPoints with me on Google Drive, while others jotted down notes in your journals. Nick V. and the Hensel girls were popular search results. Day 107. Wow! What an introduction to anatomy! We conducted an autopsy of a dead body found at the scene of a crime. Evidence included the manner of death was either homicide, suicide, or accident, and corpses had drowned, been shot or stabbed, electrocuted or poisoned. Poor little pickles. This activity has been evolving over time from the original link to accessexcellence.org. Several comments. Day 106. Creatures in Crime. Several video clips. How can protists like diatoms, plant parts like sawdust, and animals like flies help forensic scientists solve crimes? Day 105. Anatomy Outline Notes, pages 1-2, for new stampers. Keep all 6 pages with notes to exempt the midterm before Christmas break. We also watched our NHMS video with Nick and the Incredible Machine about muscles and bones. Bob is with us too. Day 104. The Anatomy PowerPoint to Rosalita. We reviewed through slides 25. We labeled Bob's bones and completed a matching activity. And yesterday I was told in the principal's office that we will be taking an exam on Thursday, 2nd and 6th, and on Friday, 1st and 5th. Day 103. I'm looking at the anatomy of a chicken wing diagram. What is the relationship among the major parts of an arm? How do they work together to allow movement? What common ancestor do birds and mammals have? Why is this significant in evolutionary terms? Are you pumped up about dissection? Monday will be a great experience. Yes! Day 102. Chicken wings are yummy, especially the tender muscle meat morsels that were extracted. What all tissue did you observe today? How do they work together to enable movement? What part of the chicken floats in the stock? Ms. Benton observed on day 102. Day 101. Bones and muscles matching review today. Some of you got to examine the dog, the armadillo, and the possum skeletons. Others were able to draw the chicken wing dissection diagram in their own style. Nice. Fifth period viewed some select x-rays that my daddy gave me. Now, what tissue shows up best on x-rays? Day 100. The universe within is a great nova about the human body. First, Mike Powell, the world record holder in the running long jump, 29 feet 4 inches, shows us the interplay among the skeletal, muscular, nervous, and circulatory systems. The brain starts it all off by stimulating the heart's pacemakers, or nodes, to contract and pump. The brain also sends an impulse to the muscles, which triggers the actin and myosin proteins to ratchet together to contract. Rob Taylor, an ice climber, broke his ankle to show us the components of blood that battle foreign invaders. Macrophages, phagocytes, B cells, and T cells all work together to destroy antigens, or germs. Now, what are antibodies? And what is the significance of our thymus gland? Several comments on day 100. And a great blood battle picture. Day 99. Today, the digestive system. Enzymes are special proteins that help other chemical reactions take place. Amylase is in your saliva, and pepsin is in your stomach. What do they break down? Which two proteins ratchet together to cause contraction in your muscles? What proteins target germs? Name the protein on red blood cells that carries oxygen. It's all about the proteins and their shape. Who remembers what luciferase does? Couple of comments. Day 98. We discussed the path food takes as it goes through the digestive tract. The stomach is the major organ of digestion, where the breakdown of molecules takes place. And the small intestine is the organ where absorption of food molecules in the bloodstream occurs. Which protein molecules accomplish this task? Where are villi found? Why are they so important? And what are the smallest blood vessels called? Today I had an observation by Connie Daniels, first period. Day 97. Clint Eller, the snake guy, was an awesome lesson for me, ×4. Each period he shared his knowledge about his reptilian pets, their ecology, adaptations, and human misunderstandings regarding snakes in general. Annabelle was a bit of a class clown, exposing herself as a central character in the presentation. Way to go, you introduced species. The selectively bred corn snake was a close second. Day 96. Language arts classes traveled to Birmingham today to witness the surroundings referenced in the novel by Cynthia McKinstry, While the World Watched. In science, we watched earthworm dissection videos and then dissected an earthworm. First period guys all did their own, while second period chose to videotape me dissecting one. Jack did his own. Fine surgery. Bella did the best job fifth period, and all day. And sixth was great as usual. Which organs store and grind food in an earthworm? And yes, came and did a fine job twice. Three comments. Day 95. Midterm exam review PowerPoint on my webpage. 50 questions. We will also take the midterm common assessment and I will record the highest grade. You may use your notes on the common assessment. Do you think this is fair? Day 94. A bit of excitement in the morning, a fire drill, and a visit from the drug dog. We had midterm exam number 1 today, and I gave out gum, which helps focus. Day 93. Midterm exam number 2, the common assessment, more gum, and a Christmas bookmark printed on color-changing paper were given for motivators. Your higher score will be posted on Infinite Campus. I hope you all have a joyful and rewarding holiday. Love to you and to your family. Day 92. Welcome back to the Kingdom of Cummings. I hope you enjoyed today's show about earthworms. Why do earthworms cover the driveway when it rains? How do earthworms move? What are some predators of earthworms? What organs of digestion do they have? How does their digestive system compare with our own? How many worms did I collect from the driveway? Can anyone find an earthworm cocoon? I will reward the first 4 students who bring one in. Day 91. Ketchup is a condiment, and catch up is a way of life in the classroom after the holidays. We located pages 1 and 2 of the anatomy outline for Jolly Rancher and looked at yesterday's blog. The earthworms are now residing in the classroom terrarium. Notes were taken on pages 3 and 4, the muscular and digestive systems, and a discussion ensued. Yup, we sang the peristalsis song and passed around the small intestine scarf. 3 digestion videos helped explain the process of digestion. Some of you drew the diagram of Emily Mathis's arm on your outline. She was quite an artist. I videotaped each class for evidence of educational value. You guys were great. 3 comments. Day 90. The circulatory system. The heart pumps blood. Arteries lead away from the heart. Capillaries are the smallest vessels. Red blood cells carry oxygen. White blood cells destroy germs. And platelets help blood clot. Now, where does diffusion take place? And what causes the heart to contract? And Justin's boo-boo was the worst, wasn't it? Day 89. We danced today. After learning about the respiratory system and the process of breathing, we got our resting pulse rate, then danced to the B-52's Love Shack to see how that would affect our breathing and pulse rate. Mine went from 67 to 85. Where does gas exchange take place? Where do food molecules enter the bloodstream? Why are they so small and numerous? Mr. Moore came to visit 5th period today. Day 88. What a dreary day outside. It was fun to be in the kingdom learning about systems of the body. specifically the excretory and nervous systems. Kidneys have nephrons that filter blood and make urine. They are tiny and numerous, just like villi and alveoli. Where are the villi and alveoli? Where does urine go after being filtered in the kidneys? What organ stores urine? The nervous system consists of the brain, the spinal cord, and neurons. Where are your peripheral nerves? Give some other words that use the prefix peri. Day 87, January the 13th, 2015. It was a Tuesday. I missed you guys today. I hope y'all treated the sub with respect. And a little bit of review never hurts. Negotiation is a dialogue between two or more people or parties intended to reach an understanding, resolve points of difference, to gain advantage for an individual or collective, or to craft outcomes to satisfy various interests. Day 86. Ah, yes, adrenaline. The hormone that gets you going. Endocrine glands are organs that secrete hormones directly into the bloodstream. They regulate bodily activities like growth and sugar levels. So whose reaction was the funniest? And who would you like to have around in an emergency situation? A couple of comments. Day 85. What a nice day. We used T-squared time to get video clips set up for class since the desktop decided to be ornery. We went over pages 9 and 10 of the outline, which was all about the senses and the endocrine system. A couple of classes heard the story of Brittany's pawpaw's guts, and Skittles were used to show the connection between smell and taste. We watched the video clips of The Scream and evaluated whose was the best response. I must say that Jack's and Olivia's responses got the highest reviews. I promise not to do that to you again. Day 84. The second 9 weeks reward day was today, and so the morning classes we watched some interesting videos about adrenaline and cameras. We also watched the squirrel going through the obstacle course. After connections, 7th graders got to pick a teacher to stay with and watch a movie. We watched Wall-E and bought drinks and candy from Ms. Ellis to support the science department. My air didn't work, so we kept the door open and some kids sat in the hall. Guess what I did Saturday? I made a video for you. Here's a link to it. I hope you enjoy. It's on YouTube. Day 83. I missed you guys on Tuesday. I hope you all treated Ms. Chester with respect. She is a former student of mine who gave me lots of great work in art. Her brother CJ did too. I love those kids. I went to my doctor today and she told me to take some time off and she prescribed some medicine for me. I want you all to do whatever the sub says and continue learning in my classroom. On your own time, you may go to my website and explore the outlines, labs, and reviews, and related files. All are linked to the standards. Send me some comments so I know how you're doing. Love to you, Miss C.